So today I will briefly introduce how we use the allogeneic hydrogel encapsulation method to make the mini ovary model and how we use this model to study the female reproductive biology and also toxicology. Okay, so our first two speakers already give you a brief introduction about the downstream female reproductive function, female reproductive tract. In our lab, we primarily focus on the gonad, the female gonad, the ovary. And there are two major functions for the ovary. At first, the ovary will produce hormones such as estrogen, inhibin, and progesterone. And the second, the ovary also mature the oocyte or egg after ovulation for maturation, ovulation, and then support the full-term pregnancy. So for the ovary, there are different developmental stages of the follicles. So today I will mention follicles many times. So for here, that is the follicle development process. So actually, primordial follicle is the earliest stage of the follicle. For women, they have 1 to 2 million primordial follicle right after birth. So right after puberty, primordial follicle will be gradually activated to the primary stage, and then further develop to the secondary, early antral, and antral stage for maturation. And during the follicle maturation process, the follicle will also secrete increasing amount of the estrogen and also inhibin. And then when the estrogen reach a peak, it will stimulate the pituitary gland to release a surge of luteinizing hormone to stimulate the ovulation of this mature follicle. So the hallmark of the ovulation include the rupture of the follicular cell and also the resumption of the oocyte meiosis. So the follicle can release a fertilizable egg for potential fertilization. For example, after the fertilization, the egg will develop to 1 cell, 2 cell, until blastocyst stage of the embryo. And at the same time, the embryo will also transport from the fallopian tube to the uterus for implantation and the placentation. So since the ovary is pretty or highly hormone regulated, it's also potential target of some toxic compounds such as clinical drugs and also environmental endocrine disrupting chemicals. So why we need to culture the ovary tissue in vitro and also in 3D? So far we know that the gold standard for studying female reproductive toxicology still rely on whole animals. But we also know that the whole animal, they are pretty expensive, time and efforts consuming, and also cost a large number of the animals. So specifically for the ovary, it is also pretty challenging for us to study the dynamic ovary function. For example, the dynamic follicle activation and the development process I just introduced, and also the dynamic hormone secretion, ovulation, and also luteinization process. So on the other hand, some other people, they use individual ovarian cell lines, such as the granulosa cells, the theca cell, or the oocyte for toxicology study or for physiology study. However, these individual cell lines do not faithfully mimic the whole function of the follicle or the ovary, because for the follicle, there's a lot of bidirectional communications. For example, for a growing follicle, there are 3 major cell types, including the outer layer of the theca cell, inner layer of the granulosa cells, and the center germ cell oocyte. So actually, the theca cell will produce androgen, which will be uptaken by the granulosa cells to convert the androgen to the estrogen by the aromatase. And also the granulosa cells and the germ cell oocyte will also talk to each other. For example, the oocyte will secrete GDF9 and BMP15, those TGF-beta family proteins, to drive the granulosa cell differentiation. And on the other hand, the granulosa cell will also provide the amino acid pyruvate to support the oocyte maturation. So this indicates if we only culture 1 cell lines in the ovary, that cannot mimic the whole function of the ovary or the follicles. So in our lab, we primarily use the allogeneic hydrogel encapsulation method to 3D grow the ovarian follicles in multiple species, including the bovine, human, and also rodent. So if we use mouse follicle as an example, we can use the allogeneic hydrogel to encapsulate the follicle, and then it can give it a 3D environment. we can culture the mouse follicle for 8 days. The follicle can grow from the multi-layer secondary stage to the early antral stage on day 4, and then to the antral stage on day 8 to reach full maturation. And then we can stimulate the antral follicle or mature follicle with the HCG or luteinizing hormone to stimulate the ovulation. And then the mature follicle can rupture and release a metaphase 2 egg or oocyte. And the remaining somatic cells will differentiate into the luteal cell, which is a process of the luteinization, and form a corpus luteum or Cl-like organoid. So if we collect the conditioned media during the culture window, we also see that actually the cultured follicle can secrete a human 28 days, like a menstrual cycle hormone secretion profile. For example, for the figure right here, the red line indicates the estradiol and the black line indicates the progesterone. And we can see the increasing level of the estrogen before ovulation, and then it will greatly decrease after the ovulation induction. And then for the progesterone, it is relatively low before ovulation or during the follicular phase, but the form, the corpus luteum organoid will secrete increasing amount of the progesterone after the ovulation. So this is the hormone secretion profile. And for the ovulated egg, we can also perform in vitro fertilization to fertilize it. And it can develop to the blastocyst stage of the embryo. And if we transfer this embryo back to the pseudo-pregnant mice, the mice can be pregnant and deliver live pups. So this data indicate, at least at the morphological level and the hormone level, we can mimic the most of the key ovary functions. So recently, we also collect those cultured follicle at different time points to perform the single follicle RNA sequencing analysis, because we want to see whether the mini ovary or the ovary organoid we cultured in vitro can mimic the key genes or signal pathways like that can support the ovary function. So for here, I won't go too much in detail, but the overall message is it can, because most of the key ovary signal pathway and the genes, they are well preserved and pretty similar to what we have been found in vivo. Just use one example. For example, the LHCGR, that is a luteinizing hormone receptor. So actually, before ovulation, the induction of the LH receptor can ensure the follicle to be responsive to the LH, and then it can ovulate. So this result indicate that our mini ovary model can mimic key ovary functions, and we can use this for studying the toxicology, biology, and also the ovary disease. So these are our current project in our lab. For example, we use the vitrification method to cryopreserve the individual follicles. In this can be used fertility preservation method for young female cancer patient, and we can also use this to make a high content a follicle biobank, so we can do the high throughput toxicology study. And we also use our in vitro ovulation model to screen a lot of compound, which is supported by the Gates Foundation. So we can do the drug repurposing and see which can be the a candidate of novel birth control pills. And we can also use this model to study the toxicology and also connect to other reproductive organ or non-reproductive organs to make it more in vivo-like. So today, because of time limitation, I will only introduce our toxicology project. So currently, we use this ovary, a mini ovary model, to screen different classes of the compound, including some clinical or the drug on the market, and also some environmental contaminant, such as the bisphenol A and the bisphenol analogs, and also the flame retardant, PFAS, nano and the microplastic, and also the cyano harmful algal bloom toxins. So today I will just use heptoxin as an example to let you know how we use this model for the toxicology study. I'm not sure whether you have heard about the cyanobacteria or the harmful algal bloom, but you may have seen some a picture, or if you go to the lake, you see some green water, or if you go to the beach, you can see some brown or blue water. Actually, those blue water are primarily caused by the cyanobacteria. And it's a great public health concern and also environmental concern. And it's primarily caused by the temperature rise due to the climate change and also the agriculture running off. So the reason we care about the bloom very much just because the cyanobacteria, they can like secrete or produce some toxins, which are also called harmful algal bloom toxins. And the microCCLR, that is the most common and also potent type of the HAP toxins. So it has been shown that it may cause like a liver toxicity, liver cancer, and it also has like a neurotoxicity. However, we still don't know whether it can be toxic to the ovary in the women or other female mammalian species. So in this study, we use our mini ovary to do the like in vitro exposure using different concentration of the microcystin. And our data indicate that, so actually during the follicular phase or before ovulation, microcystin did not affect the like overall estradiol secretion and also the follicle survival. However, what we found that after we introduce or induce the in vitro ovulation, the microcystin can dose dependently inhibit the ovulation. And for example, at 10 micromole, like less than 10% of the follicle can ovulate. So that's indicate exposure to microcystin, especially during the bloom season, like through the drinking water or through like if accidental swallow of the water, like when you swim in the bloom like a lake, it can potentially affect women's reproductive health. It may cause like anovulation, infertility, or even some reproductive disease such as the reproductive cancer or the polycystic ovarian syndrome. So for the next part, we also want to know whether we can use this model to study the mechanism. So what has been found is that microcystin is a selective inhibitor of the protein phosphatase 1 and also 2A, PP1 or PP2A. And we also know that actually PP1 is a key mediator to activate the PI3K AKT signaling pathway during the follicle like maturation. So we hypothesize that maybe that is also the mechanism that microcystin can inhibit the follicle maturation so it cannot ovulate. So using this mini ovary model, we do the same exposure, and we also found that microcystin can inhibit the PP1 activity in the follicular cells. And we also see significant downregulation of some key mediator, such as the phosphorylation of the AKT and also the FOXO1. And actually, our real-time PCR data also indicate the microcystin exposure can also significantly downregulate the like a target gene of this signal pathway. So, for example, I already introduced the LH receptor. You see, the microcystin can dramatically decrease the LH CGR expression, and that might be the reason to make the follicle not responsive to the LH surge, and then it can cause the ovulation defect. So for here, you may have a question. So all your results are based on the like a mini ovary or in vitro exposure. So whether you will see some similar or you can validate your data using the in vivo model. Actually, the answer is yes, because we also perform a like an in vivo validation experiment by using the mouse superovulation model. And you see this experiment design, and here is the result. We did consistently see that the microcystin treated mice, they have fewer ovulated eggs in the fallopian tube. And if we collect the ovary for histology, the data also show that we can see a lot of more unruptured or unovulated follicles in the microcystin treated group. And these results are consistent to what we have found using the in vitro or the mini ovary model. So actually, in addition to using the mouse sample or the whole animals, we also collaborate with IVF clinic and also validate our data using the human like primary granulosa cells, and we also see consistent data. So overall, this result indicate that microcystin or the harmful algal bloom toxin, they may have potential effect for women. Like for example, they may cause irregular menstrual cycle, or the like annual ovulation, infertility, or some reproductive disease such as a polycystic ovarian syndrome, because in like abnormal ovulation, that is a key marker of the PCOS. So for the currently, we also collaborate with Dr. Julie Kim at Northwestern University and try to make our model more in vivo-like. For example, we want to connect the ovary with downstream reproductive tract tissue, such as the uterus, so we can make a whole female reproductive tract on chip for studying the physiology and also the reproductive toxicology. And for myself, we also want to co-culture the liver tissue and the ovary tissue so we can introduce the metabolism for our reproductive toxicology screening. So overall, I hope based on this result, I can convince you that the mini ovary model is pretty cool model and also pretty powerful model for us to study the, like for example, fertility preservation, the development of novel birth control pill, and also the study the ovary biology, toxicology, and also study the whole female reproductive toxicology. So at last but not least, I would like to thank all my lab members from Rutgers University and all my collaborators from other university, and also the financial support from like NIH, NSF, and other agencies. Thank you so much.
We have time for one question.
Thanks for the very nice talk. So can you comment on the ovarian tissue concentration of HEB toxin at the serum level that you mentioned? Do we have an idea of that?
Oh, you mean the chemical concentration we use,
yes.
right? Yes.
And what is observed? So what is your expectation to see in vivo in tissues? Mm-hmm. What would be like the real exposure?
Yeah, that's a very good question. So for the chemical concentration, we did dose response concentration from 0.1 to 10 micromole. And both from the low dose to the high dose, they are pretty environmentally and human relevant, especially during the bloom season. But if there's no bloom season, for example, in our drinking water, we don't have to worry about that too much. And another concept in toxicology is about dose response relationship. Actually, we observe 90% of the ovulation in reduction, but actually based on the a benchmark dose we get is around 0.1 micromole. That is even more human relevant if for the human exposure in our environment.
Yeah, thank you. Thank you so much.